So students, in our last class under chapter biomolecules, we were discussing metabolism. We will continue discussing it. Okay, in between I will show you some pictures also, so that your understanding will be easy. So you can see there, this is metabolism. Already we have learnt what all whole processes which happens in the entire body we call metabolism. In that there are two types. This is recap of last class what we have learnt. So there are two processes what you can see there, that is anabolism and catabolism. Okay, so anabolism is, you can see there, energy is used here, energy is used and new compound is produced. That means ATP is used by the organism for synthesizing new compound. You see here, these are the new compounds. Catabolism means new compound what is there, it breaks and energy is released. Can you see the arrow mark there? This is energy which is used here to produce new compound. That is a building up process, anabolism. And catabolism, you know, it is a breakdown process where one compound here, it's broken into many pieces here. You can see it is separated and energy is released. Okay, so you can see anabolic and catabolic pathways differences. Anabolism means it's a building up process or constructive process. Simple molecules form complex molecules. You can see there all the simple molecules there, many round dot like structures, what do you see, all the simple molecules have formed a complex molecule. Then catabolism, complex molecule becomes a simpler molecule. Okay, then anabolism means it uses energy, it consumes energy. Catabolism means it releases energy. Why I used the PPT, because generally I was explaining what is energy released, what is energy used. It is better, you will better understand with the help of diagrams. So if the pictures are there, it is very interesting, so that we can learn effectively. Okay, so anabolism, for building up process, you can see that energy is used here, so it consumes energy, and here energy is released, so it releases energy. Examples you can take for building up process. Acetic acid produces cholesterol. You see there, cholesterol is newly produced, so it is a building up process. Amino acids produce protein, because long chain of amino acids, we know long chain of amino acid is a protein. So amino acids produce protein. Then photosynthesis, where food is synthesized, name only says synthesis means food is synthesized, food is produced, so it's a building up process. Example for catabolism, you see glucose breaks and produces lactic acid by glycolysis. You already know this. Respiration in plants, already you have learnt. Glycolysis means breakdown of glucose and produces pyruvic acid. Sometimes it produces lactic acid. Glucose producing lactic acid is a breakdown process where name only says glycolysis. Lysis means breakdown. Glucose breaks. Okay. Then respiration. Respiration is also catabolism, anabolism both. So it is an amphibolic process. So you can take respiration as example for both, but they have taken it for catabolism. Okay. So this is one. We will see next slide. You can see that they are showing structures. Same under metabolism only. The energy released through catabolism is stored in the form of chemical bonds. Okay. So energy released by catabolism in a breakdown process, ATP is released, that is energy rich molecule, which is stored in the form of chemical bonds. When needed, this bond is utilized for biosynthetic, osmotic and mechanical works. You know now what is meant by biosynthesis, already you have learnt in photosynthesis chapter. Biosynthesis means newly something should be produced in the living organisms. Okay, so that is C4 cycle you have learnt, which is a biosynthetic pathway, C3, C4, where ATP and NADPH are products of light reaction, which is used for production of energy, ATP. Okay, so whenever energy is required, it can be used for any synthesis process in the body, or maintaining osmotic potential of the body, or to do any physical work or mechanical work like running, jogging, all that work. Whenever energy is required, you can use it from ATP. Okay, so next You can see there the glucose, no, entering NADH, pyruvate, all that they are showing, it enters Krebs cycle. This is mitochondria where energy is produced. Oxygen is used and energy is produced. This is aerobic respiration. You have already learnt it. Then the most important energy currency in the living system is the bond energy, that is called adenosine triphosphate. What they said, energy released in the breakdown process is stored in the form of chemical bonds. No? Which chemical bond? It forms ATP. Any kind of energy in our body, it is stored in the form of ATP. Okay, that is adenosine triphosphate. Name only says. You can see 3 phosphates here. 1, 2, 3. Till now, without picture you have learnt, so you didn't get one clear idea how ATP looks like. Now you can see how clearly it looks like. See, ATP molecule, adenosine triphosphate. No, tri means 3. So 1, 2, 3 phosphates are there. That is high energy bonds formed between phosphate. Okay, so what gives energy in the ATP is these 3 phosphates only. Then it is bound to ribose sugar, that is pentose sugar. You can see 5-membered ring, carbon like 1, 2, 3, 4, 5. This is oxygen, no? Here oxygen comes, then here carbon 1, carbon 2, carbon 3, carbon 4, and here this line, what you see black line, that is carbon 5. Then it is bound to adenine. That's why it becomes adenosine. No, you know what is the meaning of it. It's a nucleoside. You have learnt already that ribose, when it binds to adenine, adenine is a nitrogen base and ribose is a pentose sugar. When it is bound to phosphate, then it becomes a nucleotide. So ATP is a nucleotide. Okay, so that is how metabolism, that is what all process happens in our entire body, produces energy in the form of ATP. Okay, then next interesting part what we are learning is living state. The living state, that is, in organisms, metabolites are present in different concentrations. You know what are metabolites? Like during the process of conversion of one biomolecule to other biomolecule, some intermediate compounds are produced. I told you in last class. Like suppose A and B are reactants. Okay, then E and F are products. A and B are reactants, E and F are products. In between C and D is there, that is an intermediate. Intermediate compound means it is not completely produced a product. In future it produces product. So it's an intermediate, like a broker in between, mediator. Okay, so in organisms, metabolites are always present in different concentration. Like there are different metabolites. I told you thousands of chemicals are produced in our body. There are different intermediate compounds. They are present in different concentration. Already you know how we measure it. In last class we have learned concentration, in what quantity these metabolites are there in the body. We measure it moles per cell or moles per liter. Like in one cell, how many moles of that metabolite is there, or for 1 liter, how many moles of metabolites are there. That is how we count. We will take example. Like blood concentration of glucose is in normal person. Blood glucose level, that is sugar level. Okay, blood glucose level. How much glucose is there in the blood of normal person is 4.2 to 6.1 millimole per liter. Means for every liter blood, for every 1 liter blood, 4.2 to 6.1 millimole of glucose will be there. Like if you take 1 liter blood, in that 4.2 to 6.1 millimole glucose will be there. That is what concentration of hormones. What about the quantity of hormones in our body? It is in nanograms. In nanograms, that is nanograms per ml. Like for every 1 ml of blood, just in nanograms, not even a gram, nanograms of hormones will be there. So what it indicates, 2 examples they have given. Some metabolites are present in higher quantity, some metabolites are present in lower quantity. That means they are present at different concentrations. Okay. Whether they are more or less, but they are required for keeping our body healthy. Then, under same heading, same metabolism, systems at equilibrium cannot perform work. As living organisms work continuously, they cannot reach equilibrium. Meaning, systems at equilibrium cannot perform work. Means, if all the metabolites are in the same concentration, then body cannot work, body cannot be normal. As living organisms work continuously, they cannot reach equilibrium. Okay. So, saturation or reaching the concentration 100% is not possible because we continuously work. When we continuously work, we will require these metabolites. So, no biomolecule will remain permanent there. One biomolecule will change to other biomolecule. That means the metabolites or the energy produced in the body cannot reach 100% equilibrium because some of them we will use, and some of them when we use, then it cannot reach equilibrium. Okay. It is like, I will give example. Suppose one drum is there. In that drum, capacity of that drum to store water is suppose 100 liters. Okay. You consider that drum as a human body. Okay. And water as metabolites. Metabolite cannot reach equilibrium. Equilibrium is, I am talking about when drum is completely full. Drum cannot be full in our house because continuously we use water from it. If we keep drinking water, it will not be full. No, it cannot reach equilibrium. We keep drinking water, so its quantity reduces. Like that, metabolites quantity also reduces. So, the living state is non-equilibrium steady state to be able to perform work. That means, if we have to perform work, then our body should not be in equilibrium. So, it is, our body should be in non-equilibrium steady state. Then only we can perform work. See, I am teaching now. That is also work only. I am doing work because my body has not reached equilibrium. So, I am in non-equilibrium state. Okay. So, living process cannot constant effort prevent falling into equilibrium. So, living system is a constant effort to falling into equilibrium. Means every living organism, every living body or living system tries to attain equilibrium but not possible. Like, we want to fill the drum completely but not possible. So, like that. So, every living system tries to continuously tries to attain equilibrium, but it is not possible. Okay, this is achieved by energy input obtained from metabolism. Meaning, body tries to attain equilibrium because of the energy released by metabolism. Okay, during catabolism process, energy is released. With that energy, body continuously tries to attain equilibrium. Because if living system should attain equilibrium, it requires energy, and that energy is released by catabolism. Okay, but it is continuously trying, but it is not possible. So, no living state without metabolism. Means, without metabolism, body cannot have a living state. That means, dead body cannot have metabolism. In dead body, metabolism is zero. Means, when the person is dead, his body will not have any processes, because all the processes in our body together we call metabolism, but dead body doesn't have metabolism. So, if the living system is there, that means there is metabolism. Means, if a person should live, if any living organism should have life, okay, that means it should have metabolism. Okay, so metabolism is compulsory. There are no living organisms without metabolism. Okay, so with that concept of metabolism we finished. Okay, so This is YouTube class, you can see this class again and again, and it is easy to understand when there is a when there are pictures. Okay. So next concept we are learning is very interesting and important, that is enzymes. We are going to learn about enzymes. Okay, so I should tell you, enzymes are biological catalysts which fastens the rate of reaction, or which will make reaction faster. I'll give example now. Let us take examples. Okay, we will take examples like any chemical process is there, any chemical process where reactants are converted to products. Without catalyst, the reaction will happen very slowly. If you want to make that reaction fast, like for example, suppose one reaction takes half an hour or 30 minutes and you want to achieve it in 1 or 2 minutes, then what you should do? You should use catalyst. Catalysts are used as a mediators, like brokers. Okay, like it is like saying, if you want to sell your house or property quickly, you cannot sell. Nowadays OLX is there, so OLX also takes time. People have to visit OLX, they have to see, they have to bargain, all that is there. But if broker is there, there are real estate agents, if you tell them, okay, today if you tell them, tomorrow only they will bring 7-8 customers, like that. Okay, whoever gives more price, you can sell, like that. So their real estate brokers are making the process very fast. Here enzymes will make reactions very fast. Okay, so that is a meaning. So what is enzyme then? Enzymes are biocatalysts, not only ordinary chemistry catalyst, because they are produced in the living organisms. They are produced in the living organisms. Like enzymes are produced by living organisms, so they should be called as biocatalyst. Bio means life or living organisms, because living organisms produce enzymes, so you should call them biocatalysts. If you are using non-living any chemical catalyst, like what you use in chemistry, like platinum, palladium, all these catalysts, these are all metals, so these are non-living, then you can just call catalyst, which is just a chemical. Here we call enzymes are biocatalysts because enzymes are produced in body of living organisms. Like human beings produce enzymes, the different enzymes. Plant also produces enzyme, bacteria produces enzyme. What this enzyme does? Enzyme hastens the rate of reaction, means fastens the rate of reaction. Like what reaction should finish in 30 minutes, it will finish in 1 or 2 minutes. So how to define enzymes then? Enzymes are biocatalysts which will enhance the rate of chemical reaction. Enhancing means increasing or making it fast. Okay, so enzymes are biocatalysts which will fasten the rate of reactions. Now one interesting statement is there, that all enzymes are proteins, but all proteins are not enzymes. Can you tell me why? In previous class we have learnt that enzymes are also proteins, hormones are also proteins. Okay, enzymes are also proteins, hormones are also proteins only. All enzymes are proteins, but all proteins are not enzymes, because some of the proteins are hormones. I hope you are understanding. I repeat, all enzymes are proteins because some of the proteins are enzymes. Okay, all the proteins are enzymes. That's why not all the proteins, some of the proteins are enzymes. So like you consider for your understanding, suppose, okay, 70% of proteins are enzymes and 30% of proteins are hormones. Now you tell me, what is the meaning of this statement? All enzymes are proteins. Yes, all the enzymes are proteins, but all proteins are not enzymes, because some of the proteins are hormones also. That is the meaning. Okay, so enzymes are very specific. Each enzyme has its own substrate, meaning enzyme is very specific. For one particular substrate, one particular enzyme is used. What is substrate means? Substrate is a material on which enzyme acts. Meaning, I'll give an example. Lactase is an enzyme. Okay, word lacta means milk. Lactase is an enzyme present in bacteria called lactobacillus, and this bacteria is present in the milk. Okay, that lactase enzyme is required for converting milk into curd. If milk is converted to curd, that is because of enzyme lactase that is present in bacteria present in the milk. Now you tell me, what is a substrate? Substrate is a material on which enzyme acts. That is, lactase enzyme acts on milk and converts milk to curd. So definitely the substrate should be milk. Okay, so any substrate, for any substrate, any enzyme you cannot use. We'll take some other example. This lactase enzyme, what is there, in the milk, it can work only on milk, not on any other material. Okay, suppose starch is there. Starch is a substrate. For starch, you cannot use lactase enzyme. You should use amylase enzyme. You see, amylase enzyme works only on starch. Lactase enzyme works only on milk. That means, I hope you understood. So enzyme is very specific. It is like lock and key model. You see here, lock and key. If you have one key, you cannot open all the locks. You can open only one particular lock. I will give example. Suppose you have your bike key, two-wheeler key or car key. Okay, with that car key, you cannot open other cars. You can open only your car, because shape of that key and the shape of the hole in the lock is exactly opposite in shape. Okay, exactly matching in shape, so that they correctly lock. I told you, like, why lactase enzyme should work only on milk, why amylase enzyme should work only on starch, because their shape of the molecule is exactly opposite. Okay, like this is an enzyme, key is an enzyme, then lock is a substrate. Okay, so we will consider this is lactase enzyme and this is milk. Milk is a substrate and lactase is an enzyme. Suppose substrate we will keep it same, milk, then enzyme is amylase, then they will have different shape, so lock doesn't open. That means enzyme doesn't work. Okay, if enzyme works, then it is called enzyme activity. No enzyme activity if you take different enzyme and different substrate. So you have to take, that is why I said enzyme is very specific. So each enzyme has its own substrate. So lactase enzyme has only milk as substrate. Amylase enzyme has only starch as substrate. Okay, so ribozymes are also enzymes. Ribozymes are enzymes where nucleic acid, RNA, that behave like enzymes. Sometimes RNA behaves as enzymes, RNA, ribonucleic acid, which is a genetic material. Sometimes RNA also acts as enzyme, then it is called ribozyme enzyme. Okay, so enzyme form tertiary structure, 3D, with some crevices or pockets called active site into which substrate fits. Okay, this I will show what is that means. Suppose this is a shape of enzyme. Okay, this is a shape of enzyme, then what should be the shape of substrate? Substrate shape should be like this, then only see it can bind, it can fit. Okay, see I am showing like this, it can fit, otherwise it is not possible, no? So you take some other shape. Suppose this is a enzyme, then I have to take substrate like this, see it fits. So suppose this is a substrate and this is an enzyme and this is a substrate, it cannot bind, see. So this is a substrate like a lock and this is an enzyme like a key. So one particular enzyme cannot act on all the substrate, only on particular substrate it acts. Okay, so like that. What they are saying, you see there in the key, one particular area is there, no, where it has a different shape, or crevices means hole. Okay, like even lock also has a hole through which key you push, no. So both of them should match. That is what he is saying. This is a pocket, and enzymes are having tertiary 3D structure with some crevices. See, some crevices means cleavage or like a cut marks. Okay, so every key will not have same shape. Keys will have different shapes. See, I will show you with different keys now. Okay, so you can see that this key is having different shape and this is having different shape. Closely you can see what kind of crevices they have. You see these cut marks, what is there, you know, these are the crevices. Okay, this is a crevice, pocket, and here these are the crevices. See, shape is completely different, you know? So this key will act on only one particular lock, not for all. And this is only for my bike, particular bike, not for any other bike. It doesn't work on any other bike. Okay, so that is what they are talking about. This particular thing, what he is showing, no? The cuts here, edges, that is called pockets or active sites into which substrate fits. That is why substrate exactly fits. If you want some more example, I will bring log and show you. Okay, I went to bring lock, so I will show you exactly. See, suppose this is a, and this is a key lock, okay, and this is a key. You can see that it has a hole here. Okay, to open this particular lock, now I am going to lock. See, this particular shape is required. I locked it. Okay, so lock is a substrate, enzyme is a key. So with one enzyme, one lock you can open. See, I opened it, but see, suppose if I lock with the different key, I cannot open, because you see this is hero bike key. Okay, with this you cannot open. It doesn't open. You see, I am pushing, but also it doesn't open. I am pushing, but also it doesn't open. Okay, so that means enzyme and substrate are very specific, especially enzymes. For one particular substrate, one particular enzyme should be used. So what this shape is there here, this is called active sites, in which or pockets in which substrate acts. Okay, so like that. That is the meaning. Okay, we will go to next page. Let us see some chemical reactions of enzymes. Chemical reactions of enzymes, where chemical compounds undergo two types of changes. One is physical change, other one is chemical change. Physical change means, you know, outer shape changes. No, some physical properties only will change. Okay, like physical change means a change in shape or state of matter without breaking bonds. Like ice melts into water, water becomes vapor. That is change of one state to other state. Like states of matter, you say, like solid becomes liquid, liquid becomes gas. Okay, gas vaporizes as, evaporates as vapor. No, and vapors after condensation, after cooling, it becomes liquid again. So it just changes from one state to other state, or change in shape. That is what we call physical change. Okay, any chemical compound you see, that is why in chemistry lab also, even in chemistry books also, first usually they give in a reactions physical test or physical reactions. Then they will go to chemical reactions. That is the exact reactions. Okay, then chemical change or chemical reaction, in this bonds are broken and new bonds are formed. bonds will be broken and new bonds will be produced. Okay, it may be organic or inorganic reaction. Organic means where carbon compounds are involved. Study of carbon compounds we say organic chemistry. Okay, then studying elements in the periodic table, 118 elements in the periodic table, that is what we call inorganic elements. So whether it is organic or inorganic, but reaction happens. For example, we'll take inorganic reaction. Okay, BaOH twice. Okay, that is barium hydroxide plus H2SO4, that is sulfuric acid, produces barium sulfate BaSO4 plus 2H2O. What you see there? What changes happened there? Ba is there here also, but OH twice, what is there? That is not there here. SO4 from this side and Ba from this side binds and becomes BaSO4, and remaining is what? H2 and OH2, that becomes 2 molecules of H2O. Okay, this is I think displacement reaction in chemistry. Displaced, no? Okay. Then organic, where starch plus water produced glucose. Okay, starch plus water produced glucose. That is one type of organic chemical reaction. Okay, then how to decide or how to determine rate of any particular physical or chemical process. Rate means speed. Okay, how fast that process happens, how to determine. Rate of the physical or chemical process is equal to amount of product produced by unit time. Okay, how fast that reaction is happening, or how much reaction is happening, in what quantity reaction is happening, that is what we call rate of physical or chemical process. Okay, ನಾವು ಕನ್ನಡದಲ್ಲಿ ದರ ಅಂತ ಕರಿತೀವಿ, ಅಥವಾ ದಟ್ ಈಸ್ ಭೌತಿಕ ಅಥವಾ ರಾಸಾಯನಿಕ ಕ್ರಿಯೆಗಳ ದರ, that is rate of physical and chemical reaction or process. Means at what speed reaction is happening and in what quantity products are produced, or in what quantity reaction is happening. That is equal to rate of physical or chemical process is equal to amount of product formed by unit time. How much product is produced in what time? Okay, like suppose 1 kg is produced in 5 minutes, then it is 1 kg divided by 5 minutes. No, like that. That is rate of reaction is equal to amount of product produced divided by time. How do we define product? We consider product word starts with letter P, so P. Unit time word starts with letter T, so we convert it to delta P divided by delta T. Same thing, amount of product produced is delta P, unit time is delta T. So rate of reaction is delta P divided by delta T, that is amount of product produced divided by unit time. Okay, so we'll go to next slide. Same heading, that is reaction, chemical reaction. Rate is a velocity if direction is specified. Means if you know the direction in which direction reaction is happening, then rate means velocity, speed of the reaction. At what speed reaction is happening. That is what I explained in last class also. Rate of the reaction means speed of the reaction. At what speed reaction happens. Then rates of physical and chemical processes are influenced by factors such as temperature. Okay, rate of chemical or physical process is determined by factors like temperature. Means if temperature is more or temperature is less, then reaction may happen faster or slower. So whether reaction should be fast or it should be slow also depends on temperature. If temperature is fixed, then rate of reaction is also fixed. No, like that. So generally rate doubles or decreases by half for every 10 degree change in either direction. Means the rate of the reaction doubles or decreases by half for every 10 degree change. Like suppose you have seen one reaction at 20 degrees Celsius. Okay, when you increase temperature to 30 degrees Celsius, then the product produced will be exactly double or else it is exactly half of it. Suppose we will take example. Suppose the product produced is 1 kg. 1 kg product produced at 20 degrees Celsius. At 30 degrees Celsius, either it should be 1 kg should become 2 kg, or 1 kg should become half kg or 500 grams. No, that is what is the meaning. Then the rate of enzyme catalyzed reactions is very high. That is why enzyme will make reaction faster. Rate means when enzyme is added, then the reaction becomes very fast. The rate of the enzyme catalyzed reaction means enzyme mediated reactions is very high. We will take example. Okay, this already you know, transport of carbon dioxide. Carbonic anhydrase is the fastest enzyme. It accelerates the following reaction in 10 million times. Do you believe? When enzyme is there. If enzyme is not there, reaction can happen only double or half. This is how many times more? 10 million times more. Okay, so carbon dioxide and water binds and produces carbonic acid, that is H2CO3. Under which enzyme? Carbonic anhydrase. Where it is present? It is present in the RBC, red blood corpuscles, and also plasma. Okay, this you have learnt in transport of carbon dioxide in respiration chapter. Carbon dioxide is transported, 70% of carbon dioxide is transported as carbonic acid, you know. No? So it is carbon dioxide with water, with carbonic anhydrase enzyme, it produces H2CO3, that is carbonic acid. Then carbonic acid, when it goes to lungs in the alveoli, again reverse reaction happens. That is carbonic acid splits into water and carbon dioxide, and this carbon dioxide will go out of the body through nostrils. Okay, so in absence of the enzyme, only 200 molecules of carbonic acid are formed in an hour. So in 1 hour, without enzyme also reaction happens, but very slow. In absence of the enzyme, only 200 molecules of carbonic acid are produced in 1 hour. Then person will die. That person will die. No, because carbon dioxide concentration will increase in his blood. Then breathing difficulty it may cause, and toxication of the body. Carbon dioxide goes out of the body very slowly. So in presence of carbonic anhydrase, about, you see there, they have given 6 lakh molecules are produced per second. Can you see the difference? What is the use of enzyme? If carbonic anhydrase enzyme is not there, in absence of that enzyme, only 200 molecules of carbonic acid is produced in 1 hour. If enzyme is there, in 1 second, 6 lakh molecules of carbonic acid is produced. That is the importance of that carbonic acid. Okay, so we will continue. So you can see that this is already you have learned, this is glycolysis steps. In respiration in plants chapter you have learned glycolysis, 10 steps. No, glucose is converted to glucose 6-phosphate, that is converted to fructose 6-phosphate, then fructose 1,6-bisphosphate, then it splits into 2, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, then it becomes 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, then pyruvate. You know, this is glycolysis. That is, glucose splits and produces 2 molecules of pyruvic acid. You already know this. Glucose splits and produces 2 molecules of pyruvic acid, because glucose is a 6-carbon compound, pyruvic acid is a 3-carbon compound. So 6-carbon compound splits into 2 molecules of 3-carbon compound. Okay, all these 10 steps require enzymes. That is why reaction happens faster. Okay, so you see there, glucose is converted to glucose 6-phosphate because of the enzyme hexokinase. Very important enzyme, you have learnt it. Then glucose 6-phosphate is converted to fructose 6-phosphate by phosphoglucose isomerase. Name of the enzyme you don't have to remember, because in the textbook it is not given. In this diagram just they are showing, just to show All these processes require enzymes, and this is not there in your syllabus anyway. So in metabolic pathway, each step is catalyzed by different enzymes. I told you in last class when we were discussing metabolism, all the metabolic reactions, any reaction which happens in the whole body requires enzymes. Without enzymes, not even a single reaction happens in our body. ಅಂದರೆ ನಮ್ಮ ದೇಹದಲ್ಲಿ ಎಂಜೈಮ್ಸ್ ಇಲ್ಲದೆ ಯಾವುದೂ ರಿಯಾಕ್ಷನ್ ಆಗೋದಿಲ್ಲ. ಪ್ರತಿ ರಿಯಾಕ್ಷನಿಗೂ ಎಲ್ಲ ರಿಯಾಕ್ಷನಿಗೂ ಎಂಜೈಮ್ಸ್ ಬೇಕೇ ಬೇಕು. Okay, so example in glycolysis, 10 different enzymes take part. See, because it's a 10-step process, for each step enzyme is required. Andre 10-step process idu, prati step igu enzymes beku. Okay. Then nature of the enzyme action that we have to see. I think that we have to learn in next class. Okay, so keep watching my classes. Thank you.